right, so I'm continuing with what we did in the last two videos, and some of this is going to be a little bit of review again. So if I'm reviewing it again, you know that it's really, really important. So what you need to remember is that there are 5 major components to this coagulation system that we're doing. Number 1 is the blood vessels. We already talked about the blood vessels and how they're constricting and how when they're exposed and torn, they release contents, right? And I want to go over that. So blood vessels, vasoconstriction, von Willebrand's factor, and collagen, exposed tissue factor, the P-selectin, the ICANS, and the PCANS. So we went over that the last videos also. Now, after the blood vessels, we have the platelets. And I want you to remember that the blood vessels and platelets, this area here is called your primary hemostasis. Okay, primary hemostasis. All right, your platelets. Now, what do the platelets do? So they come together. So when the platelets come together, they have to adhere first and they aggregate. One of the main things that the platelets are doing and providing for the rest of the coagulation system to work is the surface phospholipids. Specifically, we've got phospholipids GP1A, GP1B, 2B, 3A. So remember, we need to go back, and that was on the PowerPoints that talked about that. Okay, so primary hemostasis, vasoconstriction with blood vessels. Blood vessels release, or they don't release, but are exposing von Willebrand's factor, collagen, tissue factor. And then the platelets are providing the phospholipid surface area for the coagulation factors to come in. So if you look at the platelets, so I drew a little platelet here. Remember, on the outside, they've got your glycophospholipids and your proteins here. Okay, so this is the surface area for the coagulation factors to come in. Now, they especially love 10 and 2, but we'll get into that later. So the coagulation factors come in onto all of the surface area that the platelets are providing. So we have the coagulation factors. We also have coagulation inhibitors, because remember, we want to keep the clot localized or in a specific area. So we also have inhibitors. And remember, what we're supposed to do with the coag factors, we're supposed to get to thrombin, and then thrombin goes to fibrin, and then we have fibrin with fibrinolysis. So the 5 major components that you need to remember with coagulation is the blood vessels, the platelets, primary hemostasis, and then we have the coagulation factors, coagulation inhibitors, and fibrinolysis. So your coagulation fibers, all of this is going to be your secondary hemostasis. All right, so we went over a lot of that in the first 2 lectures and in your PowerPoint about platelets. Okay, what's really, really important that you need to know is that the platelets, so they have this surface area, and what they're going to do is they're going to attach to collagen. So I've cut my skin open and it's exposing collagen. Well, remember, they have to adhere. Well, how are they going to adhere? So platelets adhere to the collagen by your GP1A, okay? So they do that. The other thing that they're going to adhere to is your von Willebrand's factor. And they're going to attach on that, again, the exposed skin has got von Willebrand's factor, and they're going to attach to that by your GP1B and your GP2B3A. So that's why it's really important for you to remember in that PowerPoint slide about platelets about GP1A, GP1B, 2B, and 3A. Very important because that's how they're going to stick. Now, once the platelets stick, you're also going to know that the alpha granules, they do have von Willebrand's factor in them also. So once they stick initially with von Willebrand's factor, more von Willebrand's factor is going to be released by the alpha granules. And when that comes in, more platelets can come together and stick together. All right, the other thing you need to know is this von Willebrand's factor. Well, how is it stored? Well, it's going to be stored in the epithelial cells and also in your von Willebrand's factor in your alpha granules, specifically with the epithelial cells in Weevil pallid bodies. So you need to remember this term, Weevil pallid bodies. So that's how the von Willebrand's likes to be stored in these things. All right, let me see here. 5 major components. Platelets are going to stick to the blood vessel initially to collagen and your von Willebrand's factor by your GP1B, 1A, 2B, 3A. And then once those get together, the coagulation factors are going to come over with the platelets, and they're going to react with the platelets receptors and come in there. And so now we've got coagulation factors, and the coagulation factors are going to go ahead and go towards making thrombin. Now, some of the defects can happen in these receptor sites. And when we have a defect in this receptor sites, remember last time I mentioned mucous membrane, bleeding, nosebleeds, bleeding gums, things like that. You're going to have to think that we might have a problem with the attachment of the platelets. Now, 2 disorders that you have got to remember are, number 1, Bernard-Solier's, which is a problem in my GP1B, and Glasman's thrombasthemia. Glasman's thrombasthemia is a problem in 2B3A. So we could put Glasman's thrombasthemia here, Glasman's thrombasthemia there, and Bernard-Solier over here. Okay. So if I have a problem with these attachments, there are two disorders that you need to remember, and that's your Bernard-Solier and your Glasman's thrombasthemia. All right, so specifically with the, why are these important? Because especially 2B, 3A, they have receptors for fibrinogen. So over here, fibrinogen is going to want to, they have receptors for that, and if they're not working, then I'm not going to get attachment and I'm not going to get the fibrin. So this one, 2B3A, is really important for platelet to platelet attachment or aggregation. So if they don't aggregate, they're going to come off, and I'm going to go ahead and start bleeding. Okay, so again, von Willebrand's is synthesized in my endothelial cells here and in my megakaryocytes where the platelets come from, and they get their alpha granules. Okay, so when can von Willebrand's be released, right? So von Willebrand's can be released, so I don't have to just kind of get a cut there, but they can also be released if I'm under a lot of stress. I mean, sheer stress in, like, say I've got cholesterol and stuff, and I've got stress with cells coming through the blood vessels, or really hard exercise. There's some drugs, infusion of VAP, And even with adrenaline, some of these von Willebrand's factor can be released. All right. Let's see. Okay, so that is kind of a review thing of what we've already gone over. And I'm going to go ahead and do some of this erasing here so that I can go ahead and do something else. Okay. Oh, that yellow didn't come off very good. Oh, neither did that. I'm going to have to spray it. So, remind me not to use those again. Let me spray. Let's get that off. Actually, I'm going to use this. So what I'm going to be talking about next is the natural way or natural inhibitors to platelets. Natural inhibitors to, oh, it looks like it's bleeding. Okay. Oh, I did that last time too. Sprayed too much. Okay, so there are natural inhibitors. Well, before I go to the natural inhibitors, let me see if this will work. Okay, so before I go into the natural inhibitors, I want to talk a little bit about the platelet adhesion. Adhesion. H-E-S-I. Platelet adhesion. So, if I have my platelet, that's my little platelet. Okay, if I have my platelet, remember it's going to want to adhere to my von Willebrand's factor, right? Well, how in the world did it attach to von Willebrand's factor? That was with your GP1B, okay? And I'm going to have a cofactor in there, a coagulation cofactor. We haven't really talked about that yet, and that's 9, factor 9. All right, so that is how it's going to actually attach. Now remember, this von Willebrand's factor has come from the broken skin. Now the von Willebrand's factor, so this is my broken skin, I have collagen here. All right, so this collagen is from the basement membrane of my blood vessel. All right, so remember that is how it's going to go ahead and attach to that. Now remember, how did it attach to your collagen? That was the 1B. And how is it also going to attach to your von Willebrand's? This is also going to be with your GP2B3A. Okay, so I'm getting all this together. The fact that this is being attaching is going to stimulate this platelet. Woo-hoo-hoo! Now I'm stimulated. I'm very happy. This platelet now is going to be releasing your ADP, serotonin, etc. Okay, this is going to go ahead and call more platelets in order for them to come into the area. Okay, so that was just platelet. We're going to get more specific into that, but you've got to remember how the platelets are going to be adhering. Okay, All right, so we're going to look at natural. Okay, this is the natural inhibitors of my platelet function. Okay. Number one is that I've got no nitric oxide. And I've mentioned that before. The nitric oxide is going to be on the endothelial cells. I'm going to draw this out in a minute. On the endothelial cells and from your megakaryocytes and your prostaglandins. Number two is your prostacyclin. Prostacyclin. Again, where is this prostacyclin from? Endothelial cells. Where's no? Endothelial cells. Endocells. And then number three, we have a CD39. CD39 is also called ectonucleosidase. Ectonucleotidase. Okay. Well, oops, I was off camera. Okay, so what does this ectonucleosidase or CD39 do? It acts as an ADPase, and it's an ADPase. It's going to act against platelet aggregation. And so what you need to know is that all of these things are in an intact endothelial cell. So all of this that's occurring is very natural. Nothing that I have to do, no inhibitors, etc. So it's very, very natural for all of that to go ahead and happen. So my goodness gracious, aren't platelets amazing? I told you they're just so awesome. You didn't think about that when you're looking in the microscope and you're counting your cells and you got to estimate platelets. How important those little guys are. It's just truly amazing. All right, so as another reminder, the membrane of your platelet, so remember your platelet, it's got these receptors. This is made out of glycoprotein. Remember, it's a protein. Okay. The glycoprotein are the receptors, and we already said that, GP1B, 2B, 3A. Now, something that I forgot to remember to tell you is about the fibrinogen. All right, so when the platelet gets excited, woohoo, it's just getting so excited, it releases the ATP and the other granule components which we talked about. Now, the other two things that are going to be excreted here is your factor V and your fibrinogen. All right. So remember, what was the job of the coagulation factors? The job of the coagulation factors is get to thrombin. Once we get to thrombin, we're going to go to fibrin. How do I get to fibrin? I get to fibrin by fibrinogen. So you have to remember that your fibrinogen is also going to be a receptor for my glycoproteins. Okay, so fibrinogen. So over here, your GP 2B 3A, yeah, also has a receptor for your fibrinogen. So remember that. All right, now, strange thing, when my platelet is secreting my ADP. And remember, that comes from the dense granules. Your ADP is going to expose ristocetin. What? Another word I have to remember. Okay. In order for this to occur too, I have to have thrombin. Now, why is thrombin important? Thrombin is important because thrombin has got to get to fibrin, and it gets to fibrin by my fibrinogen. So you can see how all of this is so important for your platelets in order to do that. Okay, so we're going to go ahead and talk about this in more detail pretty soon. All right, your other membrane protein, your glycophorin 5A, GP5A, is a receptor site for thrombin. So you can see here, I'm releasing dense granules, I'm releasing ADP. ADP exposes ristocetin in your presence of thrombin. GP5A has a receptor site for thrombin, so again it comes over here and it gets it stimulated again, and so it perpetuates all this stimulation and aggregation of the platelets to go ahead and do that. So you know, if it's doing all this stimulation and secreting all this stuff, there's going to have to be a way for me to stop it. And yes, we're going to talk about that, but not yet. All right, so right now we're just trying to get you into the mode of how the platelets come together, how we stick together, and the importance of all of these outer surface membrane of your protein. All right, let's see here. Okay, so remember, how do all of these contents of dense granules and alpha granules, etc., get out of the platelet? Remember, they have your tubule system and your open canicular system, so that's how they do that. All right, okay. All right, how does my poor little platelet, I don't have a nucleus or anything, how does it get energy? It does have mitochondria, so that's a good. And it also has your glycogen granules for energy if it needs to. All right. Wow, that's a lot of stuff. I talked about the dense tubular system. We talked about that. Let's see. The dense tubular system in there, it's very important for it to have the calcium. Remember, we've got to have calcium for all this to work. So, if I have calcium, right, if I have calcium, I need calcium in order for this to work. So the question again is, how are your anticoagulants from your purple top and blue top, etc.? How do a lot of them work? They are calcium chelators. So I made you remember that they bind calcium. Well, now you know why they bind calcium. If you bind calcium, then all of this cannot work and the platelets won't stick together and I won't make a clot. So now you're going like, now I know why a lot of these anticoagulants are calcium chelators. All right, please, please, please go back and look at the contents of your alpha granules and your dense granules. Really important. I'm going to just list them. Oh, you have my notes anyway. So the alpha granules have platelet fibrinogen. Why is that important? I've got to go from fibrin, I got to go from thrombin to fibrin, and I'm going to do that by fibrinogen. So platelets have fibrinogen. The platelets have platelet-derived growth factor. Well, why is that important? They should probably call it not platelet-derived growth factor, but they should probably call it endothelial growth factor. But the platelet secretes this growth factor so that it heals the endothelium. They have the von Willebrand's factor. We know that. It's got the beta thromboglobulin, platelet factor 4, and your fibrinonectin. We've already said that. Now remember, all of this are in your boxes and tables in your chapter 10. So make sure you look at that. All right. Okay. Your dense granules, ADP, ATP, serotonin, and calcium. Okay. And that was pretty much a overview of what we've talked about in your first 2 lectures and going over platelets. Let's see. Okay, so the next thing, besides your endothelium having nitric oxide and your prostacyclin and your CD39, there are some other things you need to remember. All right, very, very important about our endothelium. Oh my gosh, Dr. Torres, why are you spending so much time on your endothelium? Because without the endothelium, the platelets aren't going to stick, and we're going to keep bleeding everywhere. So you have to know the really important aspects of endothelium. So endothelium has anticoagulant properties, which 3 of them I mentioned. It has procoagulant properties to help it make the clot, which I already made on another sheet. And the third portion of this that we haven't quite talked about yet is the fibrinolytic properties. Fibrinolytic properties means once the clot has been made, taking it apart. All right, so must know about endothelium. Okay, endothelium. Must know. Number 1, it has anticoagulant properties. It has procoagulant properties. Procoag properties. And it's got fibrinolytic properties. Okay, so again, let me write this better. Endothelium. All right, your endothelium must knows. Okay, again, endothelium, anticoagulant properties to make sure that there is no platelets aggregating. And remember, anticoagulant does not mean that liquid stuff in your vacutainer tubes. Anticoagulant properties. Then it's got procoagulant properties, meaning it's going to help it clot, and then fibrinolytic properties, which means it's going to make the clot dissolve. All right, so some of the anticoagulant properties we already talked about was nitric oxide, and we also talked about prostacyclin. Okay, this is a platelet inhibitor. Just remember, platelet inhibitor. Okay. It has heparin sulfate. I don't remember if I think I mentioned that once, but heparin sulfate. It has tissue factor inhibitor. Remember, procoagulant, it does have tissue factor. Tissue factor inhibitor. Protein C. Thrombomodulin. And tissue plasminogen activator. So this is used to activate fibrinolysis. Okay, so here you can see this is properties, natural properties that the endothelium has to make sure that the platelets are not going to stick. All right, the proagulant properties of endothelium, of course, are going to be your vasoconstriction, right? Tissue factor, your collagen, right? Oh, your von Willebrand's. Oh my goodness, how could I forget that? Your von Willebrand's factor. I'm going to run out of room here. P-selectin, or your P-selectin. And we also have your pecans, P-C-A-M-S, and your ICAMs. Okay, let's see, it should be about 1, 2, 3, 4, 5, 6, 7, yeah, 7, okay? And then your fibrinolytic properties, number 1 here goes back to their tissue plasminogen activator, and PA1 and TAF1, which we will get back to these when we talk, oops, can't see me. We will get back to these when we talk about our fibrinolysis. All right, so that is like really, really, really important for you to remember about the endothelium. When I start drawing the endothelium and showing how these will work so that the platelets don't make clots, this will make so much more sense to you. But for right now, you've got to remember these kind of factors that are there. And okay, I'm gonna stop this video for part 1, because when they get too long, it takes forever for me to upload them. All right, so stay tuned for part 2.